Bismillahirrahmanirrahim. My affectionate students, Assalamu alaikum wa rahmatullah. Today I am going to discuss about classification of antimicrobials. Before going to discuss about this chapter, some important features, must know component of my previous topics, that is antiviral drugs. I am repeating few important points regarding that. Pulmonary tuberculosis is an infectious disease, contagious disease, granulomatous disease, chronic disease, wasting disease. It is not a hereditary disease, and it is not, there is no inborn immunity against this disease. Immunity acquired due to TB infection and from BCG vaccination. Regarding drugs, primary drugs or first line of drugs, 5 in number. Among them, 2 is prodrugs. 2 are prodrugs. That is INH and pyrazinamide. They are prodrug. And 3 are tuberculoselective drugs. That is INH, pyrazinamide and ethambutol. They are tuberculoselective drugs. 2 are antibiotics. That is rifampicin and streptomycin. Rifampicin is a broad spectrum antibiotics, and streptomycin is an aminoglycoside. Among these 5 drugs, 4 are tuberculocidal. Only ethambutol is tuberculostatic. Now regarding DOTS, there is 4, it is divided into 6 months regimen. 2 months initial intensive therapy followed by 4 months continuous therapy. This is in case of category 1. And in case of category 2, there will be 3 months initial therapy followed by 5 months continuous therapy. Why there is initial intensive therapy? This is very important. Mycobacterium tuberculosis is a such type of organism that it will develop resistance easily against drugs if we use singly. That's why, whatever may be its efficacy, that's why several drugs used together, and in initial intensive therapy, 4 drugs should be given initially. And the objective of this using several drugs is, number 1, to delay or prevent development of bacterial resistance. Number 2, to decrease population load of the bacteria. Number 3 is to get chemotherapeutic potentiation. Number 4 is broaden spectrum coverage. And finally, but not the least, is to decrease individual toxic effect. Several drugs are used, but each drug, their dose is reduced. That's why individual toxic effect will be reduced. Now the doses of these drugs: INH 300 mg daily, rifampicin 450 mg patient having below 50 kg weight, and 600 mg daily patient having more than 50 kg weight. Then ethambutol 15 to 25 mg per kg body weight. Then pyrazinamide 25 to 35 mg per kg body weight daily, and streptomycin 1 gram daily in initial intensive therapy. And among these drugs, all drugs should be given orally, except aminoglycoside streptomycin, which must be given either IM or intravenously. Streptomycin cannot be given orally because it is a polycationic drug. It has got multiple charge. It is water soluble, lipid insoluble. It cannot be absorbed from the gut if we give it orally. It can be given orally as a gut sterilizer, But for systemic purpose, this drug must be given by injection route. And there is one point for pregnant diabetic mother, all drugs, first step drugs, can be given except aminoglycosides. That is streptomycin. And for infants below 6 years of age, streptomycin as well as ethambutol should not be given, because ethambutol will adversely affect optic nerve, adversely produces loss of visual acuity, color blindness, and infants cannot complain about regarding its blindness, regarding his shortness of vision, his loss of acuity, and ultimately he or she may lose vision permanently. That's why it is to be avoided children below 6 years of age. Now, if nursing mother or baby of a baby resides with her mother, with his or her mother, then this baby, though baby is not infective, or this baby should be, mother is active patient, this baby should be treated with INH for 6 months, single INH for 6 months. This is called antitubercular chemotherapy. Now, the most important thing is there is a relationship with glucocorticoid with steroid. We know with tubercular patient, we know TB is a wasting disease. Body resistance decreases during this period, and we know glucocorticoid also reduces body immunity. That's why glucocorticoid must not be given to a tubercular patient. But there are some situations in which it can be given for a specific reason. If a patient of hypoadrenalism suffers from tuberculosis, then he or she must be treated with glucocorticoid as a replacement therapy under the guidance of antitubercular drug. If a patient suffers from tuberculosis of ureter or urethra or pleura or pericardium or ophthalmic tuberculosis or meninges is involved, then what will happen? There will be fibrosis, cicatrization occurs, there will be obstruction. In case of pericardium effusion, there will be movement, heart movement will be interfered, lung movement in pleura will be interfered because of fibrosis, because of cicatrization, ureter may be blocked, urethra may be blocked. That's why in this situation, to prevent fibrosis, we have to give glucocorticoid under the guidance of the antitubercular drug. Must be given under the guidance of the antitubercular drug. This is all about antitubercular drugs. Now I am going to start my new topics, that is classification of antimicrobials. And there are so many ways and means by which we can classify antimicrobials. And that is according to species we can classify. That may be effective against virus, antiviral, may be effective against bacteria, antibacterial, may be effective against fungus, antifungal, may be effective against protozoa, antiprotozoan, may be effective against helminth and thelmintics. This is another way of classification. It can be classified on the nature of its effect. May kill the organism, that is cidal, may inhibit the growth and multiplication of the organism, that is static. Cide means kill, so it may be cidal. Kill the bacteria, it may be cidal, or fungus, it may be cidal. And it may arrest the growth. Stasis means arrest the growth, that is static means they arrest the growth of the organism. And they do not directly kill, but when they arrest the growth of the organism, then the bacteria, that organism may become weak, and body's natural defense mechanism will phagocytose that organism. In this way, individual becomes infection free. There are another ways of classification is according to spectrum coverage, maybe broad spectrum, maybe narrow spectrum. Broad spectrum means it covers gram-positive, gram-negative organism. They are broad spectrum. For example, tetracycline, chloramphenicol. Or it may affect only either gram-negative or gram-positive. They are narrow spectrum. Like penicillin G, sulfonamide, they are narrow spectrum. And there are another ways according to the source, maybe obtained from fungus, maybe obtained from fungus, maybe obtained from actinomycetes, maybe obtained from synthetic source, maybe obtained from fungus, as for example, penicillin, cephalosporin, glycofulvine, maybe obtained from actinomycetes like aminoglycosides, tetracycline, macrolides, chloramphenicol, obtained from actinomycetes, maybe obtained from synthetic sources, as for example, metronidazole, fluoroquinolones. These are obtained from azoles, obtained from synthetic sources. These are the various ways and means by which we can classify antimicrobials. But the most important ways by which we can classify antimicrobials according to its mechanism of action. If we consider antibacterial drug, then that can be classified according to its site of action. Number one is interferes cell wall synthesis. Number two is interferes cell membrane activity. Number three, interferes protein synthesis. Number four, interferes nucleic acid synthesis. And number four is antimetabolites. We are going to discuss these things one by one. First thing, it interferes cell wall synthesis. We know bacteria has got inner cell membrane, and there is a peptidoglycan layer and outer cell membrane in case of gram-negative organism. But in case of gram-positive, there is no outer cell membrane, but multiple layers of peptidoglycan layer. Peptidoglycan layer and outer membrane together constitutes cell wall. And bacteria, for its existence, it needs a very tough, rigid, compact, imporous cell wall, because bacteria collects its nutrients for prolonged period, and the nutrients is sugar, glucose, then amino acid, then electrolytes, especially potassium. They collected it for future use, and they are highly concentrated within the bacteria. That's why its inside osmolality is very, very high. When it comes in contact with a media where there is low osmolality, then fluid will enter into the bacteria. That's why for the protection of the bacteria, for its own existence, it needs a very tough, rigid, compact, imporous, intact cell wall. And this cell wall formation occurs by three steps. Number one is formation of building blocks. Number two is polymerization of these building blocks. And lastly, finally, the main important work is done by cross-linkage or transpeptidation reaction. The building blocks, first stage, formation of building blocks or collection of building blocks. These building blocks mainly NAM and NAG. NAM means N-acetyl muric acid, and NAG means N-acetyl glucosamine. These are synthesized by the nucleus and then goes up. This is first stage, collection of this building block. And the second stage is polymerization of these building blocks. This NAM and NAG sets in layers. It sets in layers, and where there is NAM, this NAM is tagged with 5 amino acids. On each side, maybe in up, maybe in down, maybe in right, maybe in left. Where there is NAM, it is tagged with 5 amino acids. And in this way, there is polymerization. But still, there is no compactness of the cell wall. And last stage, final stage, there is cross-linkage. There is interdigitation. And this is done by peptidoglycine bridges, cross-linkage by peptidoglycine bridge, and with the help of transpeptide enzyme. When this cross-linkage occurs, then the previous 5 amino acids, one will be dropped out. So ultimately, there will be 9 amino acid with this bacteria, with this newly formed compact cell wall. And as a result of this cross-linkage, there is interdigitation occurs, and the cell wall becomes compact. In case of gram-positive organism, there are several layers of peptidoglycate layer, and those drugs which interferes cell wall synthesis, that is beta-lactams, they will interfere the third stage of cell wall synthesis, that is cross-linkage or transpeptidation reaction. In this reaction, they disrupt the cross-linkage. As a result, there is cell wall, but it is not compact, it is not intact, it is porous cell wall. So in media where outside is low osmolality, fluid enters into the bacteria and bacteria bulges, bacteria swelled up, and ultimately the formation is spheroplast, and finally it bursts. So all that antibiotics which acts by interfering cell wall, they are bactericidal. Beta-lactams, all beta-lactams are bactericidal, and they interfere cell wall synthesis. Penicillin, cephalosporin, monobactam, carbapetam, they belongs to beta-lactam. Now if we go further inside, then they will interfere the cell membrane. Cell membrane is also important for the bacteria, and it supplies energy, its activity maintained, because there is no organelle inside the bacteria. So cell membrane functions as mitochondria, and very important role played by the cell membrane. And there are some drugs, they have got capacity, they have produced detergent like activity. They act on the cell membrane, ergosterol component adsorption occurs, and they are detergent like effect. They withdraws this ergot component from the cell membrane. As a result, membrane become leakage and the content will come out. So ultimately, finally, bacteria will die. So those interfere cell membrane, they are also cidal. Polyenes, they act in this way. Now if it go further inside, they interferes synthesis of protein. Protein synthesis occur, organism substance enter into the bacteria, binds with either 30S or binds with 50S ribosome subunit, and interferes the amino acid transfer RNA to attach on the ribosomal RNA, and thus their growing peptide chain will not form. And in this way, third group, by arresting protein synthesis, they produce their effect. But these drugs which interfere protein synthesis, they are bacteriostatic, they are not cidal, except aminoglycosides. Though they interfere protein synthesis, act by binding with 3071 subunit, but they interfere protein synthesis, but they may form aberrant protein, and this aberrant protein will cause lysis of the membrane, and the inner content will come out, and ultimately bacteria dies. So all the drugs that interfere protein synthesis, they are static except aminoglycoside, which is cidal. Now if we go further, drugs that interfere nucleic acid synthesis or function. And nucleic acid, there are enzymes, that is DNA gyrase enzyme, the RNA polymerase enzyme, the drugs that fluoroquinolones interfere DNA gyrase enzyme, and drugs rifampicin interfere DNA dependent RNA polymerase enzyme, and they interfere nucleic acid synthesis, they are and function, and they are also mostly anticidal drugs. Now there is another group is antimetabolites. Bacteria produces own nutrients, and the raw material of the bacteria is para-amino benzoic acid. From para-amino benzoic acid, bacteria produces folic acid, then to folinic acid, then DNA thymidine, then thymidylate, then uridylate, then uridine, then nucleic acid, DNA, then there will be RNA, messenger RNA, ribosomal RNA, and transfer RNA. The chain of events, flow chart is going on in this way, and in each step there are enzymes which catalyzes onward reaction. But there are some substances which acts as a false analog, like sulfonamide, due to its structural similarity with PABA, competes with PABA for the enzyme folic acid synthase for the formation of folic acid. Similarly, trimethoprim or pyrimethamine also competes with folic acid for the enzyme dihydrofolate reductase for the formation of folinic acid. So these substances, sulfonamide or trimethoprim or pyrimethamine, they act as a false substrate. Substrate is a metabolic product